So I'm Beverley, I'm the director of the Botanic Garden, and it's really nice to be here to do Science on Sundays today because it's 10 years since the very first Science on Sundays talk, which I also gave. So this whole series is my fault. The idea 10 years ago was to try to put on a series of talks that would give a little bit of access to the science that's done with the garden's collection, but in an accessible and easy way that regular visitors to the garden could enjoy. So we didn't want to put on a program of big lectures in the evening with big seminar speakers. We wanted it to be for people who would come to the garden and who could just learn a little bit more about how we use the plants in the collection. So that's what we've been doing. As I say, 10 years we've been doing it now. So I'm going to start today with a little tiny bit of history. That was one little bit. I'm going to give you a second bit because it's another anniversary at the moment. And then I'm going to tell you a little bit about why people study plants, including me, because of course the purpose of Science on Sundays is to introduce you to all the exciting things we study about plants. And then I'm going to move into my actual topic, and I'm going to tell you a little bit about pollination, how it works and why it's important we study it for food security. I'm going to explain what I mean by sexually deceptive pollination, and then I'm going to tell you how you use it to trick a fly. So that's the plan for today. And I'll start with a little bit of history. So in addition to 10 years since we started Science on Sundays, it's also 200 years since this man became Professor of Botany in the University of Cambridge. So this is John Stevens Henslow, and Henslow was Professor of Botany from 1825 to the 1840s. And it was Henslow who persuaded the university to buy this site and develop it as a botanic garden. So although we're not 200 years old yet, it took a while for that to happen. It's 200 years since the man who made it happen took up his post. Now, Henslow was really an interesting character in lots of different ways. But the interesting thing that I want to mention from the point of view of this talk series is his research. So what he was interested in was variation and species and how you could test the limits of variation and still be within the same species. And so to do that, he explored the diversity of plants, and he would look not just at one form of the plant, but he'd look at how big they got, how small, how bushy, how narrow, and try to understand how far you could stretch the description of a single species and it still be the same species. Now, he's not particularly famous for that research, but he is famous for his teaching because he taught Charles Darwin all his botany while he was here at Cambridge. So Darwin came to Cambridge to study theology, having failed at medicine in Edinburgh. And by all accounts, he didn't much enjoy the theology, but he did enjoy Henslow's botanical lectures. And so he came to all of his classes, and they would go out together at the weekend, going around the countryside, exploring plants. And eventually, it was Henslow, as Professor of Botany at the University of Cambridge, who was asked by the government to actually to be the naturalist on the voyage of the Beagle, which was going off to explore South America and the rest of the world. He said, no, he couldn't do it. And then they said, well, who would you suggest in your place? And he said, well, I've got this great student, this young man, Darwin. He'd be very good. And so it was Henslow who nominated Darwin for the voyage of the Beagle. And all the specimens that Darwin collected, so these are specimens he collected that are in our herbarium, our dried pressed plant collection, he sent back to Henslow, and Henslow stored them, and we still have that collection today of all of the plants that Darwin studied. And he knew how to study them and describe them because of those conversations he'd been having with Henslow. So we can see those in the herbarium today, but you can also see them out there in the garden. So some of the original tree plantings that Henslow put in are still there, and they're clearly designed to explore this idea of variation within a species. So these are a pair of beech trees. They're over by the policeman's hut. As you head towards the cafe, you'll find them there. There's a board that explains them. And Henslow's planted them side by side because he's confused. He wants to understand why they're both beech trees if one of them's got that cut leaf form on the left and the other one's got the round leaf form on the right. We see that in his pine trees on the main walk, on the main avenue that leads to the fountain. You'll find pine trees that are the same species, but which have different growth forms, a snow shedding form and an upright form from different parts of the world. And you can see it also in the yew trees at this western end of the glasshouse range, where there's an upright form and a pendant form. And these are all original Henslow plantings as he tries to understand this idea of the nature of variation. So it's one of the things I love about the Botanic Garden, that you can sort of walk around and see that thinking that Henslow was doing and how it led to the work that Darwin did. But actually the garden's here because Henslow was clear that to understand the plant kingdom, you needed to have access to a diversity of it. You couldn't understand plants by just looking at one. You needed to look at lots of different species. And so he persuaded the university to buy this site because he wanted the space to grow lots of different plants. And in the living collection today, we now have 8,000 species. It's always hard to put that a bit in context for people. So I we'll try two different numbers. Does anybody have any idea how many plant species there are in the world? Nobody wants to guess? Rafi does when asking you. 80,000? 400,000 actually. So actually we've only got 2% of the species diversity of the plant kingdom, but different question. Any idea how many species are native to the UK? How many plant species live wild in Britain? About 2,500. So it's actually really quite low. So we've got more than 3 times the species diversity of the UK in our collection. We've got somewhere between the flora of Congo, which is about 6,000, and the flora of Madagascar, which is about 9,000. But we're only 40 acres, so we're definitely a biodiversity hotspot. But this was Henslow's plan, that people would be able to access this living collection and use it for their research. Now, he persuaded the university that they should invest in the Botanic Garden because he wanted them to understand how important it was to study plants. And some late people, come on in. It's all right, come on in. And I just wanted to say a little bit about reasons why people might study plants, because it isn't necessarily obvious. When you hear about biology on the news, it's mostly about medicine and the sorts of applications that are important in human health and in saving lives. So when people are asked this question, or when botanists are asked this question, there tend to be 3 different answers they give, and I want to show you the 3 different answers. So one of them is about food. In order to need medicine to save lives, you need to actually feed people in the first place, and everything we eat starts off as plant material. So all of the energy in all of the food chains on the planet comes from photosynthesis. It comes from what plants can do to trap the energy of sunlight and turn it into sugar. So many of my colleagues would say that they study plants because actually understanding how plants do that and how we can improve their ability to do that is absolutely fundamental to feeding people and to maintaining the human race effectively on the planet. So that's one important reason to study plants. It wasn't Henslow's and it isn't mine. The second is that actually very often a plant is the best organism to do a piece of biology with, and that may not be obvious to people, but I just want to show you a few examples. So these are all things where a really important discovery was made first using a plant, not an animal. So most people actually know, but hadn't really thought about it, that cells, the fact that all living organisms are made up of cells, was discovered with plants. That was Robert Hooke looking at his wine bottle cork down a microscope. But actually what he was looking at was the bark of the cork oak tree. There are 2 beautiful cork oaks at the east end of the glasshouse range, if you want to go and see what a cork oak looks like. Most people do know that Gregor Mendel worked out the basic rules of inheritance and genetics using peas. You might not know that he tried to work it out using chickens. That was his first choice of organism, but it's kind of hard to persuade a chicken to mate with a chicken it doesn't like the look of. Whereas with pea plants, do what you want. And so he shifted to plants because the plant was the better organism to do the biology with. Less well known, stem cells, which you hear about in medical applications, were actually discovered in plants, in tobacco. Jumping genes, these little bits of DNA that jump around in the genome and cause mutation, were discovered by Barbara McClintock in the 1980s using maize. And since the 1990s, we've had a bit of a revolution in biology in understanding how genes are regulated and controlled. And this is through processes involving a molecule called RNA that can silence genes. And that was actually discovered in the 1990s using petunia and tomato, although very sadly they gave the Nobel Prize for it to the people who had described it in worms a year later. But there we go. Botanists always have these little minor unhappinesses in life. So sometimes people study plants because actually they're the best place to study fundamental biology. In all of these cases, the plant actually had features and traits that made it easier to do the science than if you tried to do it with an animal. The third reason people study plants, and this is my reason, is encapsulated in this picture for me. So I use this slide a lot in teaching. It's very good to get a bunch of students to ask, tell you what they think it's a picture of. And they generally say an elephant. I have had one say a rhino once, which was a bad sign. But actually for me, it's a picture of about 300 different living organisms of about 20 or 30 different species. And one of those organisms from one of those species happens to be an elephant. Now, the interesting thing is that every organism on that slide, the big woody ones at the back and the little broadleaf one here and the one with the pink flowers there and the elephant, all have the same set of problems. They've all got to get enough to eat and not be eaten themselves. They've got to get enough water, but not get waterlogged. They've got to be not too hot and not too cold. They've got to find a mate. And when they've made offspring, they've got to send them off into the world with enough resources in order to thrive and set up the next generation. Every organism on that screen has exactly the same set of problems. But the elephant has a really easy set of answers to them. It just moves. It's too hot, go and stand under a tree. There's a tiger coming, run. No mate, go and have a wander and see if you can find one. The plants, every one of them, has to solve all those problems, but they can't move. And so the solutions they have to come up with, the ways they have to ensure that they do solve those problems, are generally much cleverer than anything the elephant can do, much more interesting and much more diverse. And so that's why I study plants, because actually I'm really excited by all the different ways they solve the fundamental problems of life without having the easy option of just walking away from things. So that's my answer to why I study plants. I was going to show you an example of a system which does something really beautiful that illustrates this perfectly for me. So this is a tobacco plant, and this is moving us towards pollination, which is what I'm supposed to be talking to you about. So this is a tobacco. It's a species called Nicotiana alata. It's native to the south of the US, so Arizona, and so those sort of deserty states in the south of the United States. And it, to me, a pollination biologist, is a beautiful example of a plant that's adapted to be pollinated by moths. So moths have nice long tongues. And here's a nice long flower, and the nectar that the moth's going to drink is right at the base here, so you need a long tongue to get to it. Moths are flying at night. These flowers are visible at night because they're basically white. They actually open at night. They produce their scent at night. It's a lovely example of moth pollination. And sure enough, this is its main pollinator. It's a hawk moth. But there's a problem with this hawk moth, because as well as drinking the nectar out of the flowers and transferring the pollen around, it also lays its eggs on the leaves of this plant. And its eggs hatch into these. So this is a tobacco hornworm. And they eat the leaves. It's a caterpillar. It's a nasty, horrible, leaf-munching caterpillar. And that creates a conflict for the plant. It needs pollination, but it doesn't want its leaves eaten. And so what it does is respond to signals from its own leaves that tell it how much herbivory is happening, how much of the leaf is being eaten. And if there's too much, it changes its flowers. So it's no longer attracting moth pollinators. So let me show you how it does that. So it's a graph, and our black squares are just normal tobacco plants. Nothing bad's happening to those. The white circles are ones in which the researchers have put caterpillars. Okay, and on the y-axis is the percentage of flowers which open in the morning, which is absolutely the wrong time to open if you're moth pollinated. The flowers should open in the evening. So the ones that don't have caterpillars on, hardly any flowers open in the evening. But the ones that have got caterpillars on, as time goes by, as the number of days of caterpillars munching the leaves increase, start to open more and more of their flowers in the morning. Why are they opening their flowers in the morning? Because if they open them in the morning, they attract a different pollinator. They attract a hummingbird. And so you can get pollinated by changing the time of flowering by a different animal that doesn't come with the same problem and doesn't eat your leaves. That's really clever. This is an organism that doesn't have a brain and is rooted to the spot and is changing the time at which it produces its flowers in response to the amount of being eaten that's happening to it from a caterpillar. That's much cleverer than an elephant, much cleverer. So that's my reason to study plants. Now I'm going to talk about pollination primarily here, and I want to just explain what that is and why it's important. So pollination is basically the process of getting pollen. That's the yellow dust here produced by the male reproductive organs on the plant. It contains the male sex cells, the sperm cells. The process of getting that pollen from one flower to another flower to make seed. And it usually happens by encouraging an insect, like the bee here, to carry the pollen around for you. So the pollen contains the male sex cells. You need to get it to another flower where they fertilize the female sex flowers, sex cells, or the egg cells of the flower they land on, and that produces seed. And that's what gets the next generation. So you could have pollination of the flower by its own pollen. This pollen could fertilize this flower, but that's not very good in evolutionary terms, and most plants try to avoid self-pollination. They would prefer their pollen to go to another flower. They can't move, so they recruit the insect to do it for them. So the pollen can be transported by an insect like this or by the wind. We're going to focus on animal pollination today. And the animals don't do it just to be nice. So this bee isn't doing a favor for the flower. The bee is actually there to find something the bee wants, a reward. And the reward that the flower provides is usually nectar. So the flower will be producing sugary nectar down here. The bee isn't in the least bit interested in pollinating this flower. It's interested in feeding. It comes in to drink the nectar, but it gets covered in pollen while it's there, and then it takes the pollen with it when it goes to another flower to drink the nectar, and that pollen rubs off, and you get pollination and seed. But the nectar costs the plants energy to produce. So you have to effectively pay for the service of pollination by producing this nectar. And I'm going to come back to that because that energy that it costs the plant to produce is what triggers the sexually deceptive pollination that I want to focus on today. But just a word about why it's important for us to study pollination. Well, understanding this process and how it works is fundamental back to my colleagues who work on plants as food sources, because there are many, many flowers out there that are animal pollinated that are really important food crops. And if we can't make sure that they're properly pollinated, then we're limiting the food supplies that we can get. And this doesn't apply to the cereals that form most of the calories in the human diet, so wheat and rice, maize. These are not animal pollinated, but it does apply to an enormous number of food crops. And those food crops come from, in my mind, in two main categories. There are things like oilseed rape and legumes, which are actually quite good at making seeds without animal pollination. But nonetheless, if they do have animal pollination, they make more, 10% more, 20% more. And when you think about the amount of oilseed rape or beans of different kinds that are grown across the planet, that equates to an enormous amount of human food resource that's dependent on animal pollination. And then my other category of food crops that require animal pollination are these rather more pretty flowers around the outside, which are all things that are absolutely 100% dependent on animals for pollination. So no animal pollination, no food. And going around the side in order, that's Brazil nut, top left. That's a cucurbit, doesn't matter which one, melon, cucumber, squash, they're all 100% dependent on animals for pollination. That's vanilla orchid, has to be hand pollinated when it's grown in Madagascar as a crop because there's no native pollinator. And most important of all, bottom right is chocolate. It'd be very sad if we didn't have any of that. But these all rely 100% on animals for pollination. And so understanding how that pollination works is really important for the diversity of food that we all enjoy. So there are lots of different types of pollination out there, and understanding how they work feeds into all of these conversations. The type that I wanted to talk about today is some work we've been doing on what I call sexually deceptive pollination. So I need to explain what I mean there. So this is the slide I showed you before where I explained that the bee here is looking for a food reward, and that food reward is nectar, and that costs the plant energy to produce. And so actually, that creates a selective pressure. If you're a plant who can cheat and not produce nectar, then you're ahead in the game. You're going to get pollinated without spending your energy making a reward. So if you can do it without paying the pollinators, so much the better. And there are various different ways that plants cheat in this game. I wanted to show you 3 of them. So the first is what we call simple food deception. The flower on the left is an absolutely standard flower, makes nectar. Bees come, they drink the nectar, pollen gets transferred, just like I've shown you. The flower on the right, which is an orchid, grows in the same habitat, looks very similar, and doesn't make any nectar at all. And so what happens here is that bees learn that these are nice flowers, nice and rewarding, full of yummy nectar, and they see this and think, oh, that's pretty much the same, isn't it? And they go to that, no nectar, and they do the pollination. There are disadvantages to this system for the flower, but it's pretty clever. And it's not the only example. There are lots of examples of food deceptive pollinations. This is a South African example. The lily on the left makes plenty of nectar, the orchid on the right doesn't. So food deceptive pollination is one way that flowers cheat. Second way that they cheat is carrion deceptive pollination. So this little thing is mimicking a piece of dead meat. And what it's attracting is the sorts of flies and beetles that like to lay their eggs on a bit of dead meat, so that when those eggs hatch, the larvae, the maggots that they produce, can eat the dead meat. So carrion beetles, dung beetles, this sort of thing. And so we have colours that mimic rotting flesh, reds and yellows. It will produce scents that mimic rotting flesh as well. They smell quite disgusting. They often have hairs on them to mimic that kind of mammalian skin texture. Our favourite of these, we have multiple in the collection, but our favourite, of course, is the Titanarum, which some of you may remember flowering. I think we last flowered a Titanarum in 2017. So we must be getting quite close to flowering one again soon. So not only do we have the colours, and that, again, interesting colour effect and mottling colouring effect of rotting flesh, but the really clever thing about this plant, photographed with a thermal camera here, is that the tip here is 37 degrees centigrade. It heats itself up to the body temperature of a mammal to increase that mimicry and also to make sure the horrible smells it produces are spread throughout the rainforest. So this is really impressive. No nectar, getting pollinated through trickery. The type of trickery I wanted to tell you about, though, is sexually deceptive pollination. And this is where, rather than looking like another flower that has got nectar or looking like a piece of dead meat, you look like a female insect. And you attract a male insect who thinks he's found a mate. And while he's there, he gets pollen on him. You don't have to give him any nectar because he wasn't looking for food, he was looking for a girl. So these are two different species of Ophrys, which is a genus of orchids, and they mimic the females of different insects. You can see here, for instance, we've got hair, we've got the blue sheen, which mimics the particular sheen on the wings of the wasp that pollinates this one. This one's got two lovely eye spots here that go with everything going on. Really very clever. And particularly clever because it's very hard for a male animal to learn not to visit what it thinks is a female animal. That's not an evolutionarily successful strategy. And so it's very hard for the animals not to do. So here's an insect visiting that orchid I just showed you. It's the orchids here, the insect's on top. And while he's here trying desperately to mate with this flower and wondering what's going wrong, the pollen, which is here, is being stuck on his head. And so when he goes to the next flower, it rubs off, and that's how we get seed without any need for nectar. So that's sexually deceptive pollination. A very clever system. Some of these orchids actually produce not just the colours and visual mimicry, but they also produce scents. They can produce molecules that look very much like the sex hormones of the insects to actually trigger responses by scent as well. And it was thought that sexual deception only happened in the orchids. But I've been working for the last 10 years or so on a completely different system, which is a daisy that also tricks male insects by producing mimics of female insects on its flowers. So here's my daisy. It's called Gaultheria diffusa. Here's the male fly, and here are the mimics. So we have these spots on the flower that look like female flies. And this is a purely visual system. There's no scent involved at all. Daisy does it all by how it looks. So it's a great system to work with, Gaultheria, because it has all sorts of interesting properties that make it a great model for me. Primary one is there are lots and lots and lots of different varieties of this plant. So here are 16 different versions of it. It's all one species. It's quite a narrow range. It only lives in South Africa and Namibia, but nonetheless, I can find populations with really very different flowers. And that gives me the opportunity to compare things. I can try and work out, well, you know, this one isn't making any sexually deceptive spots. So what's different about that one compared to this one? This one is not doing a very good job of sexual deception. It doesn't convince anybody. So what's going on with that one? So it gives me a really good set of tools to work with. And it's a great plant to study because you get to go to amazing places. So it's what we call a winter rainfall annual. It lives in the north of South Africa and the south of Namibia in essentially desert areas. And when the rains come, which is about July most years, then the habitat is completely transformed and all of these annual daisies pop up. So this isn't all my daisy. There are lots of other daisies in there too. But it's a fabulous environment to go and work in and to see these things in action. If it doesn't rain, nothing flowers. That's very sad when it happens. So all of those varieties, which we call morphotypes, have the same pollinator. And that pollinator isn't a bee, it's a fly. It's a type of fly called a bee fly, just to confuse you, but it is a fly. And I want to show you how that fly interacts with the flower. So on the left is one of the varieties that isn't sexually deceptive, just looks like a daisy. So the fly comes in, lands, has a little look around in the middle, there's a little bit of nectar, it's not very exciting, has a drink, flies off. The one on the right is sexually deceptive. So here comes the fly. Now he doesn't just land anywhere, he positions himself quite carefully. That was the female he thought he saw from above. And he's fairly sure there was a female here. So he's going to stay, he's going to hang around. He's having a little bit of a wiggle and a jiggle and trying to work out where she's gone. And because he's a bit confused, he stays much longer, and I'm going to have to keep talking because unlike the other one, which flew off quite quickly, he spends twice the amount of time on the flower. And the pollen is produced by this central area. And because he spends twice the amount of time on the flower, he gets more pollen put on his head. And so it's a much more effective strategy to get the same fly to pollinate you. He spends longer and you don't have to give him any nectar because he thinks he's found a female. So this is the system we've been working on. Now, I do just need to explain one tiny bit of botanical jargon to you. Some of you may know this, I imagine most of you don't, But if you don't, this is your botanical piece of knowledge for the day, which is that a daisy isn't a flower. When you look at a daisy, including your standard garden daisy, you've got lots and lots and lots of tiny flowers in the middle. In a garden daisy, these are the yellow ones, and they're all little round radially symmetrical flowers, and each one of them has reproductive organs and produces pollen and can make seed. And then around the outside, you've got a different type of flower, which we call ray flowers, and they've still got the reproductive structures. It's still a whole flower, it's not a petal, but it's got this mad, crazy, elongated petal thing going on. And if you put them all together, you get this kind of mimic of a whole flower. So what's happening in our daisy is that some of these, but not all of them, are making the fake flies. And that's what we've been trying to work out, how they do it. So we spent a bit of time, this is my colleague Alan Ellis at Stellenbosch University, who I work with on this system, thinking about who was pollinating and how they were pollinating. And we discovered that all of these different varieties of daisy have the same fly pollinating them. But what the flies do differs by variety. So on the chart here, black means tries to mate with the flower. Grey means is confused. And pale grey, white means just feeds. And you can see that some of them have a lot of sexually deceptive phenotype, and some of them have much less. And we were able to actually pin that down and characterize the spot, that deceptive spot, of the different morphotypes and relate it to the percentage of sexual deception we see. So 80% of the fly visits to this plant here are sexually deceptive, and none are to this. And we can see that what makes you sexually deceptive is a number of different traits that I'm going to talk about in a bit more detail. So we could work out what was important in the spot. And fundamentally, it comes down to three things. So the one on the left is not sexy, the one on the right is. And it comes down to three things. One is the color. So the one on the right has actually green color here. And that dark green pigmentation mimics the dark green exoskeleton of the fly. Second thing it comes down to is a chunk of texture, which you can just about see here. So there's actually multicellular papillae or bobbles on the surface of the petal in the sexually deceptive one. And those give a kind of reflectance effect that mimics the wings, but they also give a tactile effect that means the fly stays longer. He thinks he can feel something. Is that a girl? It's a bit strange. It doesn't feel like a flower. So that's quite important in the sexual deception. And the third is the switch from having a ring of spots, like the one on the left, to having these odd isolated spots. And that's really important because nobody's going to be convinced that there's a fly sitting on every ray floret. That doesn't look like fake flies. That does. So you need to have a switch so that only some of them produce it. And so in the last few years, we've tried to work out how you build each of those three things. And we've done that using an experimental technique called RNA-seq, which I just want to explain because it's something that's used a lot in biology these days, but it's perhaps not something you've come across before. So the idea is we're interested in which genes in the daisy are making those traits. And the way that genes work, you may remember this all the way back from school, is that they are transcribed into a molecule called RNA, and then that's translated into proteins, and the proteins do the job. And so in this RNA-seq analysis, what we've done is take some of the plain ray florets and some of the ones with the spots, and we've mushed them up, and we've extracted all the RNA molecules from both of them. So that's all the genes that are active in this one, and all the genes that are active in this one, in different tubes. And then we compare those sets of RNA. And all the stuff that's the same in them both is about making a petal and making orange and photosynthesis and respiration and all the things that the plant has to do to survive. But the things that are different between them should be about making the spot, because the only thing that's different between this and this is a spot. They've come from the same individual daisy, okay? So that's the idea of RNA-seq. Now, if you actually did it with the fully mature flower that I've shown you here, you would find genes that were about the business of being a daisy, not about growing the spot. So you actually have to go back in developmental time to the stages of the plant where those things are being built if you want to find the genes that are about building them, which is what I want to do. So when we first started this work, the earliest stage at which we could be confident that a ray floret was going to have a spot was this stage here, which I call stage 6. You could just see the dark pigment beginning to form. And so we harvested petals at this stage, ones that were going to have the spot, ones that weren't going to have the spot, and compared them. And I'm going to tell you about the genes we found. that affect those three different things that make the spot sexy. This is how the data look. It's a bit scary, but don't worry. Essentially, everything on this side of the zero line is active or more active in the spots, and everything on that side is more active not in the spots. And so then you compare. And we've color-coded here because we picked out genes that were involved in traits that we thought might be interesting. So the first thing we found was a bunch of genes that are about making red pigment. So every gene name in red here is a gene that was active in our spotted tissues. And together they form a pathway that ends up producing this molecule here, which is called a malonylated cyanidin, which sits in the vacuoles of the cell and is red. Okay? And we proved that by taking the gene at the top of the pathway, sticking it in a tobacco plant, and the tissues that should normally be green go red. So hurrah, we can make red. Except that our fly spot isn't red, reddy purple. It's green. We need to get this green. So we can sort of see that we have got this reddy purple pigment, but we haven't explained why it's green. Going back to this crazy data set, there's a set of genes in orange here that are involved in moving the metal iron around the plant. And we thought, well, that's odd. Why is iron being moved around the plant? Is there more iron in our spotted ray florets than our non-spotted? Well, here's non-spotted and here's spotted, no difference. But if we take the spotted ray floret and divide it in half, there's hardly any in the tip, and it's all in the base where the spot is. Okay, we thought, so there is lots of iron in the spot. We could have checked that much more simply by actually just staining for iron. There it is in the spot. Didn't know that till we'd done all the difficult work. And what's iron doing? Well, in this pathway I just showed you, again, all of these genes in red are upregulated in the spot, and what they do is push iron into the vacuole of the cell where the anthocyanin is, and in combination, a red or purple cyanidin and iron produces a blue molecule. So now we have a blue metallocyanidin in those spot cells, which is why we can see bluey colors around here. And if you take a blue pigment and put it on this yellowy orange background, you get the green, which is what's making the color. So now we know how you make the color that makes the petal spot sexy. Then the texture. Well, these are some genes here in red, which really confused us because they're involved in making root hairs. What are they doing in a petal? And we picked one of them, an expansin it's called, and we thought, well, that's funny because these papillae on the petal, they are big and chunky, and you need to kind of grow the cell big to do that. And these expansin genes in the root hairs do this little job of, they're an enzyme basically, they break the bonds between the cellulose and the hemicellulose in a plant cell wall, and that means the cell can get big. And normally, in normal plants, they do that in the root hairs. So that's these little hair structures coming off the root here. And by doing it in the root hairs, they make those hairs grow out and you get more surface area for uptaking water and nutrients from the soil. This actually is our gene from our petal system, where it's expressed in a root. And you can see that we've got it in the root hairs. So this is a case where a gene that in most plants is involved in making root hairs has suddenly found itself active in the petal in this plant. And it's making those bobbly effects that makes the male fly think it's a female fly. And the third question was about the position. So how do you end up with just one or two or three and not all of them? And the genes we picked out that I wanted to mention on this are involved in flower initiation. And it's this gene here called SPL1. So we had some idea which ray florets are going to be the spotted ones. It's not random. If you start with this as the first ray that develops, the second one that has the spot will be about 137 degrees round the plant from it. And the third one will be about 137 degrees around from that. And if you get a fourth one, which you do sometimes, it would be up here. And this is actually the spiral pattern that plants use to determine where to put their leaves and flowers normally. What's happening essentially is that the spotted rays are the first ones to develop. So there's a spot signal and then it gets turned off. And so we thought, well, maybe this SPL1 gene is the spot signal, because actually what it does is tell a plant normally to make flowers. So this is a normal plant and one in which we've expressed our SPL1 and it makes it make flowers quickly. So we thought maybe it had changed so that rather than saying make flowers, it was saying make spotted flowers. So we had this sort of model where we had SPL1 on, and on, and on, and then some sort of repressor turning it off so that in the fourth position and so later you wouldn't get the spots. What might the repressor be? That took us a long time to figure out. But there are these molecules called small RNAs, microRNAs, which turn off genes. And when we looked specifically for those in our data set, we found we had a batch of them that were strongly expressed in the non-spotted ray florets. And we could tell from their sequence that they would downregulate our SPL1 gene. So our model now looks like this, that we have our SPL1 gene saying, make spotted rays, make spotted rays, make spotted rays. And then this microRNA gets switched on, which stops it. So no longer get any spotted rays. And that's how we get the transition. We think this is still a kind of working model. I've just got a whole batch of data this week that turns that back to front, which is very annoying. I'll tell you what we did. We expressed the SPL1 gene at a very high level in a plant that normally has all of its rays spotted, and I thought it wouldn't do anything because all of the rays are spotted, and actually it's turned off the spots. I've got to try and figure out that and how it's working. So that's our model at the moment for how you do it. But there's a problem with all of this, which is this stage where we sampled is even though it looks early, is already probably too late because the pigment's already there. So this thing already knows it's going to be a spotted ray floret, and I wanted to know how it knows it's going to be a spotted ray floret. So I have an astonishing, amazing postdoc who worked out by looking at these, and these are tiny, these are 100 microns across, looking at these primordial flowers as they develop, which of the rays are going to be the spotted one, which you could work out based on their position because they're the first 1, 2, 3 to develop. And so we have now repeated that RNA-seq experiment with stage 5 and stage 4 and stage 3. And I have some very exciting genes in there that I'm going to tell you about in a couple of years' time if you come back and listen again. So I'm going to finish. It's not a finished story. The idea is that you see the work we do with the collection. It's all ongoing, it's happening. But I do need to thank the people whose work I've talked about. So everything I do is in collaboration with Alan at the University of Stellenbosch. The RNA-seq experiment was done by Roman on the left there. He's a Swiss postdoc who's now moved back to Switzerland. And in the lab, Adaya here at the back is a fabulous microscopist who's worked out which are the ray florets that we need to target. And Farah here has done all the work on the pigment that I've explained to you as well. We have lots of kind people who give us funding, which I also appreciate. And I was going to finish by just advertising the next few talks in the series and hope that you might come and hear some more as well. So thank you very much for listening today.